Good evening, and good morning, everyone. My name is Dr. Rohit Bhatia. I work as acquisition editor with Thieme Chemistry. I welcome you all for today's webinar on process chemistry to methodology, development in metal-mediated chemistry and catalysis. So here are a few rule keepings for today's webinars. Today's webinar is being recorded. The audience will be kept on mute during the web seminar. Please ask your question and answers in a chat box, and we will take as many questions as possible in live after the presentation. You can tweet also about your experience and question by tagging us at the rate Thieme Chemistry on if you are present on X. You can subscribe to our Cheminar series to have recording and watch these seminars recorded at the Cassini's website. And also you can subscribe our newsletter to know more about our forthcoming webinars and our publications. So let me introduce you to Thieme. So Thieme is a German company which has a presence of 130-year-old publishing company, which has a presence over all around the globe. We are present in major continents, and we have a following products in our portfolio. We are basically more concentrated on organic chemistry, as you know. So we have Synthesis and Synlit, which are our legacy journals, which are about 6-decade-old journals. Synlit is 3-decade-old journal. Then we have a SYNFACT, which is a highlighting journal. Then we have open access journals, SynOpen and organic materials. We also have a databases, SOS Science of Synthesis. This is the one and only curated database for organic chemistry, which is far different from the other abstracting databases. Then we have a pharmaceutical substances, which is database for the active APIs substances. It has 3,200 API listed in, which have all the information from synthesis to regulatory to the patent. Every information that you want is on this platform available. Recently, this year, we have launched another new journal, open journal, Sustainability and Circularity now. And this is a journal which is being launched under the leadership of Professor Chris. So if you are working in sustainability and circularity, we are open, and there are no submission APCs for the manuscript submitted till 31st October. So you can submit also. Further, we are also recruiting reviewers for our select crowd review for sustainability and circularity. If you would like to review with us on select crowd review, kindly email us your CV, sustainability and circularity now at @thieme.de. And now the question is, what is select crowd review? Select crowd review is a revolutionary reviewing system which is fast, fair, and very helpful. So in this, we normally try to get you the peer review back in 4 to 5 working days after the submission of manuscript. Here, the author submits the manuscript. Editor invites the crowd review members. They assess journal requirement and scope. If it doesn't meet, it rejects. If it's met, the crowd review editor share it with the crowd. Crowd is the people of the peer reviewers, high quality peer reviewers. They give their comments. Crowd editor collates them and then decide whether it need revision or rejection with review or article is accepted. So here this system works very fast, as there can be more than two people are commenting in on your paper instead of the traditional two people. So you will get review faster back, and the rate of publication, time of publication will be faster. So if you like to join us and participate for our Syn journals as a select crowd reviewer, kindly email us your CV to synthesis_crowd@thieme.in. So this is introduction for ours today's product line and the select crowd review for Thieme. Now I just like to introduce you to the first speaker of the Cheminar. The first speaker of today's Cheminar is Dr. Vittorio, please. And just a moment, please. So Dr. Vittorio is a full professor of organic University of Turin in Italy. He received his PhD in chemical science from University of Madrid in
activation is responsible here for this initiation period. I will skip the NMR slide, and I would like to move on to show you that we have extended the scope also to triflates. We found that they are more reactive than chlorides, and we wanted to look into them because with triflates we can access different structures, because we can generate triflates from phenols, which are quite abundant, and also we can generate alkenyl triflates from ketones. So we could do the reaction under exactly the same conditions, and we could show that triflates perform better with some substrates which were really bad, with some structures which were really bad with aryl chlorides. And then, yeah, we could show that we can functionalize phenols and ketones too. We then moved to more bulky triflates, because these were really challenging substrates, and we wanted to couple bulky triflates with bulky thiols. But this did not work with xanthos anymore, so we did a quick ligand test, and we found that more flexible, slightly less sterically hindered ligand, DP-EFOS, is better to couple bulky triflates. So we could show, after optimizing the reaction a bit more, that this is true for very, very different functionalities, from different functional groups. And I think in sake of time, I will skip the mechanistic discussion. You can read it in our publication. But what we also did was that we could synthesize the supposed oxidative addition complex. So we started from nickel COD this time, and then prepared this nickel zero complex, and then did the oxidative addition. And yeah, we got the crystal structure of the triflate, and we found that this was actually a cationic complex. And this cationic complex could be converted in stoichiometric reaction in the corresponding thiol, but it could also be used as a catalyst. So this shows that this is an active species within the catalytic cycle. And this is my last scientific slide now. So I show you that we have made this triflate complex. And here on this slide, I'm showing you the reported procedure, how to make the chloride complex. And this is very typical one, how to access these complexes. You start from ligand nickel 2, and then in second step add the Grignard reagent. So it does then transmetallation in the second step. The complex has two geometries, therefore we also see two signals in NMR. But the problems which we have with the synthesis, it was a nice one, but we could only access certain structures. And in order to access more structures, we needed different procedure. For example, we wanted to have different halides here. So my PhD student has developed a new way how to make them, again starting from nickel 2 and ligand, but using a reductive approach. He could obtain this nickel zero precursor on multigram scale, and then this was quite active in oxidative addition of a different array of aryl halides. And then we could also exchange the X against other anions, and we are now investigating these complexes in catalysis, and indeed we found out that triflates are by far the best ones. Well, and with this, I would like to sum up my talk. I have shown you two stories from our research. First example where we use thioesters as substrates in couplings, and second example where we make thioethers, both by using non-pressure metals catalysts. And for this, I would like to thank my co-workers, the previous ones and also the current ones. I thank my sponsors, and I thank all of you for your kind attention.
Thank you, Professor Ivana, for your nice talk. And we have a few interesting questions in the chat box, so I have collated them. So the first question is, can we synthesize thioesters from beta-nitrostyrienes?
In the thiocarbonylation, yeah, nitro functionality, I don't remember, but usually nitro functionality is problematic and it often doesn't work. Yeah.
Okay. The second question from the same person is, if there is a poor overlapping, I don't know, like they were saying, like if there is a very poor overlapping, then why need palladium or metal catalysis for further functionalization? I don't know.
Yeah, that's a good question. Certainly, the thioesters would react with some organometallic reagents also on their own, but the problem would be which you have with typically with carboxylic acid derivatives.
I think there is.
At the end, you get.
Professor Ivana, we lost you.
Where at which point?
At the start of your answer, we lost you.
Okay, so the problem in the absence of the catalyst is that you have over addition of the organometallic reagent and you produce the alcohol. In the presence of catalyst allows you to do the acyl substitution very efficiently at mild conditions and to control the reactivity. Over addition does not happen.
Okay. So the next question is, how does the organomanganese reagent compare to organozincs in terms of hydrolysis and in terms of nucleophilicity?
So they are more reactive and they are not as sensitive, but of course you have to be careful. And the problem is that in presence of oxygen, for example, they form peroxides. So usually you make them and you have to use them directly. You cannot store them.
Okay. So the next is interesting question. Alkyl translation metal compounds are prone to beta hydrogen elimination. So why doesn't this occur in this case? So I don't know. This is from.
Yeah, with
this is a question from Dr.
Yeah, and that was meant in the coupling with organomanganese reagents. I think that it can happen, and so we only know that somehow the sulfide stabilizes the iron too. But I'm sorry, we could not study the reaction by EPR because it was very complex. So we don't know much about this. So the reaction is quite quick. If we do it for longer, we can also observe the beta hydride elimination. That can happen. Yeah, so sorry, I don't have much more mechanistic information on this.
Okay, so there were like another question. Thank you for interesting talk. Maybe I missed this. You mentioned that the thioester is more electrophilic, but why it shows slower hydrolysis compared to the esters?
I don't know if the hydrolysis is slower compared to esters, but it's quite slow compared to other nucleophiles. And this was quite a lot studied by people who investigate the development of life on Earth. And I only remembered from the studies that it's somehow a kinetic phenomenon, not a thermodynamic one. But why, I unfortunately don't know.
Okay. So there is one last question we are taking. Can you tie the coupling of S-silyl aryl triplets instead of aryl triplets?
No. We didn't. We didn't try this. It's a good idea. Thank you.
Thank you, Professor Ivana, for your nice talk. I think Professor
Thank you.
Matthew has, you can stop sharing, please. Professor Matthew, as Dr. Matthew has joined us back. Dr. Matthew, can you start with your presentation?
Sure, we'll go ahead and pick this up again. Sorry about the technical difficulties earlier. Rohit, can you see the slides now?
Yeah, we see Dr. Matthew.
Okay, so yeah, changing topics again, we're coming back to drug discovery and industry. And we were saying before that this is a process that has many stages leading from target validation and hit finding all the way up to clinical development and commercial release for stability. And when you're doing this kind of work, you can have many questions along the way. At an early stage, then you have the strong interest in exploring biological features faster through structure-activity relationships, SAR, and structure-property relationships, SPR. And it's possible to leverage automation in order to do that faster and get novel compounds faster. And the focus of today's talk is going to be highlighting examples from successful case studies and discovery process chemistry collaborations. And when we say discovery process chemistry, what we're really talking about is accelerating compounds from the medicinal chemistry part of the chevron into the process chemistry part of the chevron, which is essentially an acceleration process that we think about happening through two ways. On one hand, discovery process chemistry collaborations focus on developing chemistry that accelerates medicinal chemistry SAR interrogation. And on the other hand, we have process chemistry facing objectives, which are to develop scalable, fit-for-purpose routes for both key intermediates and also clinical candidates. So these are sort of the examples that we try to think about when we talk about discovery process chemistry. And these are the kind of examples that we highlighted in this short review that was published in Synthesis around a month ago, shown here on the right. In general, discovery process chemistry, or DPC, shown at the center of this cube, is really an intersectoral area of innovation. And when I say intersectoral, I'm talking about the intersection between pharma, academia, and CROs. On one hand, the pharmaceutical industry gives a strong point of innovation in the form of drug design, as this is really the source of where many new drugs are being designed through collaboration and advancing many different programs. Academia has a role to play in this, though, which is to really advance the frontiers of current knowledge in terms of the chemical space that we can access with synthetic organic chemistry. And CROs, or contract research organizations, which is in a large part where a lot of the large-scale execution of structure-activity relationship, or SAR, interrogation takes place, as well as the scale-up of lead compounds. So this is really the intersectoral space that characterizes discovery process chemistry. And the key message that I want to be able to deliver to you in today's talk is that really discovery process chemistry is about implementing the latest synthetic chemistry methodologies in organic synthesis in order to accelerate SAR and SPR, as well as early process development. And as we start to go through some of the examples and we see how this can work in practice, if you are someone in the industry, then I'll try and show you how Eurofins Discovery can work together with you to advance your programs through discovery process chemistry collaborations. So to get started, I thought we could highlight some case studies from the literature, and there'll be case studies from discovery process chemistry. Some of these will be case studies where we see how it's possible to accelerate SAR or SPR through the use of bioisosteres. Later, we'll talk about the possibility of using couplings of diversifiable reaction partners. And then towards the end, we'll go into examples of late-stage functionalization. After this, we'll also take a look at some examples of internal case studies from Eurofins Discovery. But I want to give fair credit to all the examples that have already been published in the literature so far. Taking a look at the case of bioisosteres, first let's introduce this general concept. So here on the left, we can see an example of an aromatic ring, which we often see presented to us in drug substructures, whether building blocks or final API candidates. And there are a number of different bioisosteres that we can imagine taking the place of a simple aromatic ring in order to modulate the properties. And that could mean either the physical chemical properties, things like solubility, things like metabolic stability, but it could also mean modifying the potency of the compound. And as an example of this, here on the right, we have BMS 708-163, which was modified with a bicyclopentane bioisostere motif in order to improve the permeability as well as the aqueous solubility. Talking about some examples of how BCPs can be installed and how this can be used in principle to evaluate the SAR and SPR of biorelevant scaffolds. Here are some examples from a collaboration between Pfizer and the Anderson Group at the University of Oxford. In this first example, we have a primary alcohol being transformed through an Appel reaction in this first step into an alkyl iodide, which can then be used to difunctionalize a propylene in order to arrive at alkyl iodides, which are quite useful motifs to have installed in a BCP core. In the second example, then, this is another example of how the same type of alkyl iodide can be used as a precursor for installing a BCP iodide, here in the context of sugar chemistry. And when you can install BCPs in this type of very complex framework, then you know that this is chemistry that can be potentially applied to survey the SAR or SPR in a program with a lot of power. In some other examples developed in the schema and the scope of this collaboration between Pfizer and the Anderson Group at Oxford, then there was another difunctionalization of propylenes in the form of this photoredox example, where radicals could be generated from alkyl iodides added across the central bond of propylenes and trapped with iodine in order to form these types of scaffolds. And here, this is another radical mechanism similar to the ones on the previous slide, where a large number of scaffolds could be tolerated. And this is quite an important feature when we think about identifying reactions.
Dr. Matthew, your audio is not okay. So we wait for some minute for Dr. Matthew to rejoin.
He's rejoined, Roy.
Okay.
Okay, apologies for that. Hopefully it won't happen again, but it looks like the internet is not so stable today. Okay, let's come back to this. So we were talking about examples of installing bioisosteres, specifically bicyclopentanes, in drug-like scaffolds. Here's a pretty concrete example of how bioisosteres can be used to compare biological activity and physical chemical properties of drug-like scaffolds to other non-bioisosteric motifs. For example, in this example from GSK, on the left we have a bicyclopentane which has been synthesized using the methodology on the top through a decarboxylative radical generation, followed by a Manishi functionalization with heteroaromatic cores in order to install this type of heterocycle. And by comparing that against the aromatic counterparts, it's possible to see what is the effect of installing that BCP core. And the conclusion that we can arrive at is that it's possible to have a higher degree of solubility, aqueous solubility, that is, with comparable levels of intrinsic clearance here at the bottom, that's CLint, as well as the artificial membrane permeability. So that means it's possible to tune one property while keeping others constant, which is quite an important ability to be able to have in medicinal chemistry. And then on the next slide, we'll have a look at another example of bioisosterism in practice. Okay, we keep dropping off. Hopefully it's fine now. Rohit, can you still see the slides?
Yeah. We can see
Yes.
your slide.
Okay. Yeah, apologies to everyone who's watching and for the technical difficulties. So here we're focused on accessing one bioisosteric compound from the precursors of another, which is to say, how would you access a 1,2,3 triazole using the precursors of an amide? The solution that the authors came up with here was to use carboxylic acids, which are precursors to amides, in order to access alkynes, which are precursors to 1,2,3 triazoles. And the solution that they came up with for this interconversion is to use a decarbonylative cross-coupling between carboxylic acids and alkynes, using a bimetallic catalysis system using palladium and copper, shown here. And using this system, then they were able to unite carboxylic acid fragments and alkyne fragments and form the type of products shown here, which also had a large degree of functional group tolerance, particularly in the case of aromatic heterocycles, which is, of course, something you want to see for a methodology that you're trying to apply in the context of drug discovery. Here's another example of a discovery process case study, which I wanted to look at in some detail. And this is one which is less about bioisosterism and more about coupling a diversifiable reaction partner. Specifically, we're talking about coupling this nitroalkene up here on the left with an azomethine imine precursor shown just to the right of that. And the reaction of interest here is an asymmetric 3+2 cycle addition to arrive at a tetrasubstituted pyrrolidine product using a chiral ligand bound to a copper catalyst. And this is quite a powerful reaction as we're forming four contiguous stereocenters. And when you think about how to apply that kind of convergent reaction in the context of a drug discovery campaign, you can imagine that being able to change every single piece of those components would really allow you to look at a wide range of chemical space. And in this example, then that's exactly what the authors did. They changed every single component. They looked at a huge number of analogs. In this case, over 1,600 analogs was prepared using this methodology, which really allowed them to look at a wide range of chemical space and optimize their SAR and SPR very quickly. Additionally, these authors were then able to apply this methodology in the process chemistry group later, where not only was the reaction useful for SAR and reaction and drug discovery, but also for the development process. Here's how it was applied in the context of the downstream development. In the first step, glycine could be condensed onto 2-tolualdehyde. Afterwards, they were able to apply the azomethine imine precursor with the asymmetric 3+2 cycle addition that we had a look at earlier. And then through a 3-step sequence involving a Bach protection and an F reaction and reduction of a ketone diester selectively, it's possible to arrive at another tetrasubstituted pyrrolidine, which could then be used to form the tetrasubstituted pyrrolidine core of ABBV3221. So you can see sometimes that when pharma companies are investing in finding reactions that survey the SAR faster, they're also identifying solutions that are going to be useful for downstream development. Here's another example from the Baron group in BMS. Quite interesting, the now probably well-known cross-coupling reaction of redox active esters with dialkyl zinc using the catalysis of nickel with these BIPI ligands. This is quite a useful fragment when it comes to forming carbon-carbon bonds between aliphatic fragments, particularly when you think about forming carbon-carbon bonds between aliphatic fragments originating from carboxylic acid derivatives, because carboxylic acid derivatives there's a huge number of them in the literature and in the commercial feedstocks. And these examples here in the bottom left, these are cases where in the collaboration, they were able to develop the chemistry and functionalize these types of drug-like molecules or drug molecules at a late stage. And then the top right, we have a case where in the same publication, the authors were able to functionalize a carboxylic acid located on a peptide on a resin through the formation of a redox active ester, a cross-coupling using the methodology in the top left, and a deprotection with cleavage off the resin in order to form a peptide bearing a natural amino acid. In the bottom right, we also have an example of how the authors were able to do a 3-component coupling using the same type of activated carboxylic acid, these redox active esters, which is just adding more diversity to the number of things that you can make using this type of approach with redox active ester chemistry. And finally, transitioning to thinking about how late-stage functionalization can be used to probe SAR, I wanted to highlight one example from Patrick Fear and Merck. This is chemistry where, sorry, we'll come back. This is chemistry where the authors developed an amino functionalization of pyridines at the 2 position. And this is a chemistry which takes what I think you could call a reagent-based approach to late-stage functionalization, where this reagent here, bearing the amino group shown in orange, was used in excess, slight excess of 1.5 equivalents in order to functionalize the pyridine. The proposed mechanism is going by initial attack of the pyridine on the reagent to displace a chlorine and form a pyridinium, followed by cyclization of the pendant amine onto the pyridinium in order to result in a dearomatization, and then a rearomatization by deprotonation with a base, as the rearomatization would then release and rearomatize the pyridinium, which can then undergo the cleavage under the treatment with zinc acetic acid in the second step in order to result in the aminopyridine product. And here are some examples of the kinds of very functionalized pyridines you can use with this kind of chemistry. And this is the approach I wanted to highlight because I think that when you have these kinds of reactions that do functionalization of specifically aromatic heterocycles at a late stage, these are incredibly valuable in pharma. So if you're in academia and you're thinking, what reactions should I be developing? These are examples I would highlight as things that are useful for industrial chemists. Another example from late stage functionalization that I wanted to show comes from a collaboration between the Levin Group and Janssen. This is using what is probably well known to many of the members of this audience, this anomeric amide developed by the Levin Group. And here, the authors were showing that it's possible to react amines with this anomeric amide and extrude the amine with concomitant installation of a bromine, hydroxyl, sulfide, or phosphonate. So you can reach actually a lot of different products using amines, which are, of course, very present in a lot of both drugs and also precursors to drugs and intermediates to drugs. So in this specific mechanism, what they're proposing is essentially that the amine reacts with the reagent in order to form this type of Zwitter ionic intermediate, which in the case of the bromination late-stage functionalization reacts with the tribromomethane radical in order to do a CH abstraction to form this intermediate in the southeast, which can then decompose with release of nitrogen gas in order to form a carbon-centered radical, which can then abstract bromine in order to form a brominated product. And in the publication between the industry and academia, then here's amines being converted into hydroxyl groups. And in the bottom, we have examples of amines being converted into phosphonates, or also thiols. So yeah, this is another example of sort of reagent-based late-stage functionalization. Also a very useful approach to late-stage functionalization for SAR interrogation. Because you can also imagine that if you can turn if you can turn an amine into one of these groups, then you can also convert these into these functional groups, bromides, hydroxyl groups, into quite a lot of other things additionally. So it's really multiplying the amount of chemical space that you have access to. And then one last example of late-stage functionalization that I wanted to show is shown here on the slide, which focuses on chemistry of Kotritsky salts. So in this first reaction on the top left, in the deaminative functionalization via reductive cross-coupling, this is a reaction which is going under the catalysis of nickel with BIPI ligand. As well as cleavage and cyclization, was used by Merck in their PC SK9 program in order to form this macrocyclic peptide, which could then undergo a late stage reductive cross-coupling in order to selectively install the Kotritsky salt in the bottom left-hand side of this molecule, and then convert that Kotritsky salt into an aromatic and heteroaromatic fragment in a very controlled way. So you can imagine that it would be very complicated to try to form that one specific derivative if you had to go through the whole solid phase peptide synthesis from a non-natural amino acid. And if you wanted to make new derivatives, you'd have to go through the whole synthesis again, of course. So that's a really useful approach, this late stage functionalization approach, that is, when we talk about formation of new solid phase peptide analogs. And with that, I want to show briefly a couple case studies on completely unrelated chemistry that have come out of Eurofins discovery. In the first case study that I want to show, this comes from a collaboration with Janssen, or J&J. And this is a case study that's focused on Alzheimer's disease. So it turns out that there's a particular post-translational modification of the tau protein, or specifically installation of O-linked N-acetyl D-glucosamine, which is linked to lowering protein aggregation associated with Alzheimer's disease. And the key enzyme implicated in this process, or OGA, can be inhibited in order to essentially provide a therapeutic route for Alzheimer's disease. So a medchem campaign was started using the compound on the left side of this arrow at the beginning of a hit to lead medchem campaign. And essentially by varying the heteroaromatic portion on the part, the central scaffold in the middle, and the nitrogen substituent in the southeast, it was possible to optimize the structure further. In the bottom left, we have an example of how the structure activity and structure property relationship were interrogated. This is a sort of simple representation, but in one case, you can see how one nitrogen in the spire cycle can undergo an SNAR while the other one's protected. And then by subsequently deprotecting the other nitrogen, it's possible to carry out a second functionalization, which could either be through SNAR or cross-coupling, for example. In order to reach more complex and more diverse scaffolds at the spire cycle, it was also possible to develop diversity-oriented chemistry focusing on the spire cycle itself. So in this box in the bottom right-hand corner, we have pyrrolidine bearing an ester, which could be used to reach the type of iodo-allyl fragment, the second intermediate on the right. And that could be then used in a palladium-catalyzed borylative cyclization in order to form a 5,4 spire cyclic core bearing a B-pin, a pendant B-pin fragment. And that pendant B-pin fragment is actually very useful if you think about reaching out to new chemical space from a complex new scaffold, because you can imagine doing a lot of different Suzukis. You can imagine parallelizing that kind of process and reaching a lot of different new IP space by taking that type of approach. And so these are the approaches that were applied in this program. And in the end, the orally bioavailable OGA inhibitor shown here on the right was formed. And a second case study I want to show, this one comes from Bayer. And in this case, we're focused on coronary artery disease. And it seems that there's a disintegrin in metalloprotease with thrombospondin type 1 repeat 7, or ADAMTS7, which is linked to the development of coronary artery disease. And it turns out that selective ADAM-TS7 inhibitors may have therapeutic potential for treating CAD. So this is sort of the motivation for targeting this one specific enzyme, ADAM-TS7. And through a virtual library screening, this initial starting point shown here on the left side of the arrow was identified. And that starting point had significant ADAM-TS7 inhibition, but it also had ADAM-TS5 as well as ADAM-TS4 inhibition. And ideally, we wanted to be even more specific. So from the hit of that virtual library screening, a hit-to-lead medchem campaign was carried out using the chemistry in the bottom box. So in order to interrogate the SAR in detail, the chemistry team used what's called a Bucherer-Bergs reaction, which essentially involves converting ketones into alpha amino ketones and then reacting it into form hydantoin cores with concomitant formation of a stereocenter with a pendant amine. And that protected pendant amine can then be deprotected and install a variety of different substituents, including, which are just represented here as an orange ball, and essentially interrogate the SAR in this way. And it was this sort of approach that allowed the identification of what substituents need to be placed at that very congested stereocenter on the left. And through the hit-to-lead medchem campaign, then once again, an orally bioavailable candidate was selected. And with the few minutes I have left here, I just want to talk about how chemistry collaborations work at Eurofins Discovery. So yeah, in the previous talks we've heard from today, then these are primarily academic talks, but since I have the chance to speak to what I think is primarily an academic audience, I want to give a little bit of insight into what the world of chemistry CRO looks like. And here in Eurofins Discovery, we're focusing on a wide range of scales, leading from milligram to kilogram. With compounds or procedures that are primarily known, either in academic literature or in patent literature. And here in Eurofins Discovery, we've also co-located ADME services in order to do our primary assays in the same place that we do the chemistry labs. And here we've also installed a robotics lab so that the early phase can be accelerated by parallelization. Here in Eurofins Discovery, our chemistry teams, which are highlighted here in this picture, are working closely with our scale-up laboratory, as well as the ADME team, the high-throughput experimentation team, and also our high-potency lab, in order to allow them to basically interrogate SARs in a lot of different types of programs with whatever tools they need. So that could mean, in the case of HTE, that they are doing parallelization in order to make a lot of different analogs of one drug scaffold. It could mean, in the case of the scale-up lab, that they're working with a scale-up lab to produce a diversifiable intermediate or early non-GMP tox batches for developability studies. Or it could mean that they're doing work in the high-potency lab with PBOEL 3B or 4 compounds. So that would be the highest potency categories. And beyond chemistry, Eurofins Discovery is also doing things including hit finding, in vitro pharmacology, safety pharmacology, and ADME-PK, as well as a lot of other kinds of support for drug discovery programs. And in order to essentially be able to support lots of different aspects of the drug discovery space, then we have a lot of services in the same place, which allows us to do essentially chemistry support for lots of stages of drug discovery, ranging from comp chem all the way up through the HT work that we saw earlier, to the ADME assays, to high throughput, high containment, scale up, and to be able to co-locate all of these in one central European lab here in Murcia, Spain. And beyond chemistry, then Eurofins Discovery also does what's called integrated drug discovery. And that allows them to essentially support lots of different types of drug discovery programs that could be a small molecule in nature, or that could be protease targeting chimeras or antibody drug conjugates, ADCs, using computational chemistry, using computational tools, using artificial intelligence predictions of what ADME properties will be, using synthetic chemistry support, using robotics and automation, and using scale-up. And in short, yeah, that's just a little bit about Eurofins discovery there at the end. But in general, the main message from today is that the field of discovery process chemistry is really about shortening the critical path to drug discovery using methodologies from recent literature and enabling SAR and SPR as quickly as possible, as well as enabling the improvement of process development along the way. And if you're in the industry, or also if you're in academia and you're interested in exploring partnerships, feel free to reach out. We do all kinds of different partnerships that are focused on a lot of different areas, and we are always open to different discussions about ways of working together. And with that, I would end. And thank you again for your patience with the technical difficulties today. You can feel free to reach out about anything at all by email through the email here, or you can also find me on LinkedIn if you want to chat about anything at all. And I'd be happy to answer any questions if anyone's still around.
There are plenty of people still around, and we have questions also, Matthew. So we have two very good questions also. Basically, this is a very great presentation. As academicians or a lab chemist, and transition and thinking to the process chemistry is a good challenge. Like I have done myself while I was doing that reaction in flask and then trying to mimic that reaction at 100 kg pilot plant. It's a way more challenging than what we face in lab. So that's a really eye-opener examples and case study that you have shared with us. So there were two very good questions we have here. So the first question is from the process perspective, what are the advantages and disadvantages of using nickel and zinc in reactions? Even at lab scales, preparing active nickel is not easy. On the other hand, I have seen some reaction that produces zinc salts after the reaction, but even at a lab scale, these produce viscous and a fine-grained salt that are difficult to filter.
Yeah, so I would say for sure there are reactions which you would call discovery reactions, there are discovery process reactions, and there are process reactions. And it could be that the fastest way to discover the drug in the next drug candidate, or to discover and survey the SAR, could be to use one reaction that you would just never use if the drug goes forward. It could be that the fastest path to making a carbon-carbon bond could be a photoredox coupling using a very expensive iridium. And in the med chemist's mind, he knows that you will just never go for that system if you decide to scale it up later. Maybe it will just be easier to use a Suzuki, but it might be the fastest thing at the moment for whatever reason to survey the SAR using this method. So there are sort of fit for purpose solutions, and then there are best process solutions. And as the drug becomes increasingly or advances towards a candidate declaration, then you start transitioning from fit for purpose solutions to best process solutions. And a pharma company will increasingly devote more and more resources to refining with increasing granularity the process that's being applied. And
Thank you, Matthew. Another problem that I saw like in process chemistry is waste management of these active compounds, which are generated at very high scales. That is another process management challenges that we have. That is for some other time. So there is another question. So the person is asking, I would like to ask the role of zinc in amination of pyridines. Could you tell more about this, please? Because the mechanism you showed did not mention about zinc. Thank you.
Sure, I'd have to look at how that cleavage reaction is working. But yeah, my assumption is that if there's a pyridinium, then my assumption is that the nucleophile will attack the carbon linked to the pyridinium and hydrolyze the pyridinium carbon bond. I don't know whether that nucleophile will be acetate or something else. I don't know what the role of zinc is, to be honest. I mean, I guess sometimes you see carbon-nitrogen cationic nitrogen bonds being cleaved by zinc, and then so I guess I would assume that this is going by oxidative insertion and then protodemethylation. I'd have to check about that exactly, though. That's a good question.
Thank you, Dr. Matthew. I think there are no more questions, and if you have any questions, you can still post them after we were sharing this recorded version on our Cassini platform. the questions there. We try to answer them. I thank you all the speakers, all the three speakers, for a very interesting talk and giving us perspective how we can use metal-mediated chemistry from process to the lab chemistry. Thank you very much, and we can closing this webinar. Thank you very much, all of you, for joining us for this interesting webinar.